Hello everybody, welcome to Ms. McGuire online video lecture. Today we're covering cell division, mitosis. So let's go ahead and begin. So after completing these chapters, you will be able to describe the main events of the cell cycle, indicate how chromosomes change and interact during mitosis, discuss control of the cell cycle, and list the events of apoptosis. Cell division and death. An adult human body consists of about 30 trillion cells. New cells form as old one dies at different rates in different tissues. Growth, development, maintaining health, and healing from disease or injury require an intricate interplay between the rates of mitosis and cytokinesis and apoptosis. Now, let me pick up my pen. Now, mitosis is the division of the nucleus and division of DNA. So when your cells divide, they need to divide their genetic material equally between two daughter cells. Of course, if we're talking about mitosis, right? So cells will divide their genetic materials, chromosomes, equally between two daughter cells. And then cells need to divide cytoplasm. All those organelles and fluids inside the cells need to be distributed equally between two daughter cells. So that's what cytokinesis is. And apoptosis is a precise, genetically programmed sequence of events that leads to cell death. So how your body function is actually depends on these three processes. Mitosis is cytokinesis, so that's a division of cells, and death of a cell. So you don't want too many cells die, and you don't want extra cells to be formed. That's why this need to be interplay and between those rates of cell division and cell death. So in this picture, they show you mitosis and apoptosis. And you can see during mitosis, we go from fewer cells to more cells. So cells divide and we make more cells. This is how your organism grows. This is how zygote, that is the first cell that gives rise to your entire body. Zygote divides, forming billion of cells that make all your organs and your body parts. And at the same time, apoptosis, you go from more cells to less cells. In this example, syndactomy is a genetic condition when apoptosis fails to separate fingers of a baby. So first, during our fetal stage, when we're still inside mother uterus, our fingers are actually connected. And through apoptosis of cells between the fingers separate, right? So if we don't have this precise synchrony, precise rate between mitosis and apoptosis during embryonic development, people can born with some birth defect, like syndactomy or connected fingers, like in this example. Before talking about cell division, we need to understand what a cell cycle is. A cell cycle is a sequence of events associated with cell division. So cell cycle represented by a pie diagram, and it's divided into several phases. So shown here in blue is mitosis. So at the beginning of mitosis, right? Oh, I'm sorry, at the end of mitosis, when we have cytokinesis, new cell is formed, right? So we form a new cell over here, and then cell goes through the series of ordered events until the cells divide again, right? So cell cycle includes a major part is called interphase, right? So all this without blue part is interphase. When cell is growing, performs its functions, replicates its DNA, pretty much as cells does what it's supposed to do and preparing itself for division. And it includes G1 phase, S phase, and G2 phase. And then cells enter mitotic phase when cells divide. Some cells do not divide, then they enter what we say G0 phase, and either the cell just remains specialized, so just specialized cells that carry on its function, or it can proceed to either cell division from G0, or it can die and never divide. All right, but that's a cell cycle, and we will look at different phases in more details. So stages of the cell cycle: interphase. It's a preparation for cell division. During interphase, cells need to replicate all their genetic material, so replicate DNA and also subcellular structures. Interphase composed of G1S and G2 phases. Cells may exit the cell cycle at G1, right? So at G1, it exits and enters G0. It's a quiet phase. So when cell is in G0, that means it's not ready to divide yet. Chromosomes are replicated during interphase, and it's done before mitosis. So it's prior to mitosis. Results in each chromosome consisting of 2 copies joined at centromere. So when chromosomes replicate, when they make copy of themselves, then they made of 2 copies, right? They made those copies, so they're made of 2 copies. And these 2 copies hold together at the centromere. Proteins forming mitotic spindle is synthesized, and microtubules from centrioles near the nucleus start growing those mitotic spindles, getting ready to, well, they don't grow in interphase yet. It just centrioles, I'm sorry, I'm not gonna go ahead of myself, so we will say that what happened in interphase, we replicate chromosomes, we synthesize proteins that will form the spindle, and we will form centrioles, right? That's all happen in interphase. So here's interphase. Chromosome are not condensed. So here's a cell nucleus shown in blue. This is nucleolus. This is the area of the nucleus where proteins are assembled and ribosomal RNA is synthesized. But if you look inside the nucleus, you don't really see any chromosomes. It doesn't mean that you don't have genetic material here. It just, the chromosomes are non-condensed yet, so they're in a loose form, and under light microscope, you cannot see it. So the best characteristic of interphase when you're looking at a microscope slide of cells would be nucleus without chromosomes. So chromosomes are uncondensed. Now, why chromosomes are uncondensed? Because remember, in interphase, you need to make copies of those chromosomes. And you cannot make copies of chromosomes if they are in very tight, packed, condensed shape. So the chromosomes must be loose, uncondensed, so you can make copies of them. So when chromosome is not replicated, it's made, so it's shown over here, it's made of chromatin, and it's not condensed yet. But after DNA synthesis, now this chromosome shown over here, it's replicated already. And it's made of 2 identical copies that called sister chromatids. And they hold together in an area called centromere. And remember, this happens before mitosis. So it happens in interphase. And actually in a specific phase of interphase called S phase. And S stands for synthesis. After chromosomes are replicated and, some other processes happens within the cell, we will talk about them in a minute, like cell need to pass some checkpoints, but if everything is done correctly, then cell will enter mitosis. And the first part, the first stage of mitosis is prophase. During prophase, DNA coils tightly. Chromosomes shortens and thickens. So chromosomes became shorter and thicker. Then we start formation of microtubules into spindle. Now, spindle is a special structure that will allow your cells to separate chromosomes. So here we have the beginning of spindle formation. Now, chromosomes are inside nucleus, so we need to break nuclear membrane or nuclear envelope. So chromosomes can enter cytoplasm and they can be split between two daughter cells. So nuclear membrane breaks down and a nucleolus is no longer visible. So here the slide of cell in prophase. So in the nucleus, you can see now those lines, those are chromosomes and they condense. So we can see now distinct chromosomes. And this is a picture that shows you a cell. Those are centrioles. Centrioles are organelles that help your cell to divide. And those lines are spindle fibers. So those are special proteins that will help to separate sister chromatids and kind of like pull the cells, two daughter cells, away from each other. And inside the nucleus, you see already duplicated condensed chromosomes. So prophase, condensed chromosomes take up stain. That means you can make them visible. So you use stain to give color to the cells, so you can see different parts of the cell. So chromosomes take up stain. The spindle assembles, so spindles are those proteins. Centrioles move apart and they appear. So you see how they're on the different poles. And the nuclear envelope begin to break. So it's break down at the end of prophase. So here we still see nuclear envelope, but it will disappear during prophase. After prophase, we have metaphase. Chromosomes attach to the spindle at their centromeres, and they align along the cell's equator. Metaphase chromosomes are under great tension, but they appear motionless because they are pulled with equal force on both sides. So let's see what exactly it means and how it looks on the histology slide. So here our centrioles, all right, those are the spindle, mitotic spindle, and chromosomes right in the middle. So here's the picture. Again, we don't see nuclear envelope anymore, so there is no nucleus. Centrioles moves even further to the poles of a cell, and right in the equator of the cell, we see how chromosome line up, right? So chromosome line up during metaphase. So I remember it as M for middle, so chromosomes in the middle. And it seems like chromosomes are not moving, but actually they pulled apart with equal force. That's why they appears motionless. So that's a metaphase. Next is anaphase. So plasma membrane indents at the center. Centromeres divide. Remember the centromeres that hold sister chromosomes together? Now, when they divide, that releases the tension. Then chromatids separate and become independent chromosome, and they start moving to opposite ends of the cell. During this very brief phase, a cell frequently contains twice the normal number of chromosomes. So only during anaphase, for a short period of time, your cells have double number of chromosomes. Instead of 46, they have 92 chromosomes. So let's see how it looks on the slide. You can see centrioles, right? You can see spindle, and now look over here. Your chromosomes are separating. Now, what we're separating? We're separating sister chromatids, right? So each chromosome is made of two sister chromatids. When they are separated, we call them chromosomes. So anaphase, centromeres part, and chromatids separates. So centromeres divide, right? And they release the tension. So now the protein, spindle protein, contract and moving sister chromatids away from each other, distributing half of chromosomes in one cell and half in a second daughter cells. So I remember it, A anaphase, chromosomes are moving away from each other. And then we move to the last stage of mitosis, telophase. The cell looks like a dumbbell with a set of chromosomes at each end. Spindle falls apart because we don't need it anymore, right? Why do we need spindle for? To separate sister chromatids. When we've done our job in anaphase, spindle now falls apart. Nuclei and membranes around the nucleus reform, so we have nuclei again, and they reform at each end of elongated cells. Division of the genetic material is now complete. So now let's see how we done with mitosis. After mitosis, remember, we will have cytokinesis. But at this point, you start seeing like two daughter cells, right? So almost, they're not separated yet, but you can see this, what is called cleavage furrow. You can see that chromosomes arrived to the daughter cells. Nuclear envelopes start to be start reforming again. So we have nuclear envelope, and you see nucleolus and no centrosome. So telophase, the spindle disassembles and the nuclear envelope reforms. So that's distinct characteristic of telophase. Now, DNA is separated equally between two daughter cells. So we finished nuclear division. Now we're moving to cytokinesis. Actually, cytokinesis or division of cytoplasm begins during late anaphase. So everything is continuous process. You don't have end of one process, beginning of another. Lots of processes happens at the same time. So your chromosomes start moving to the poles of the cells, and at the same time, we start separation of cytoplasm. But we do have a special term to describe division of cytoplasm, that is cytokinesis. So during cytokinesis, cytoplasmic division occurs after nuclear division is completed. Organelles and macromolecules are distributed between the two daughter cells. Microfilaments band contracts, separating two cells. Now we have this microfilaments between, it's a special protein between two cells. So when those microfilaments contract, two daughter cells are separated. Okay, so and that's the end of mitosis. Now everything must be controlled. So okay, before we get to this point, let's look one more time what we accomplished so far. So we will just move to cell cycle, and we will see that we started mitosis, right? Started as prophase, metaphase, anaphase, telophase. So that's a division of nucleus. And then we separate all the organelles, all the macromolecules, all the fluids between two daughter cells, and we call it cytokinesis. And again, remember, cytokinesis does not begin at the end of telophase. It starts at the end of anaphase, right? So cell is divided, so the end of mitosis. So we go from one parent cell to two daughter cells. But of course, this process must be regulated. And that's why we have control mechanism. So control of mitosis is a daunting task. Too little and an injury goes unrepaired. So if you don't have enough mitosis, if you cannot divide your cells and you have injury, for example, you cut your cells, so some cells are dead, you need to replace them with new synthesized cells. So if it's too little, then injury goes unrepaired. But if too much, then you have abnormal growth that you call tumor, and some tumor can be cancerous. So that's why you have groups of interacting proteins that function at specific times in a cell cycles, and group of proteins called checkpoints. So we have checkpoints proteins. They ensure that chromosomes are correctly replicated and apportioned into daughter cells. So we need to replicate chromosomes, and we need to split them equally between two daughter cells. So that's why we have those proteins called checkpoints. Internal and external factors can affect a cell's mitotic clock. That means it can affect when cells divide, and can they finish their division, or they can never divide really and will die. So let's look at these checkpoints. Right, so here we have several points here. So let's say we're gonna start trying to get my pen. So we're gonna start right here. So imagine we have a new cell, and this cell is ready to divide again. So it will enter G1 phase, and then so it will grow bigger. It will perform its cellular functions. And if cell is really, if it does not enter G0, that means it will proceed and replicate its DNA. So right here in the S phase, DNA is replicated. And that's why we have over here DNA damage checkpoint. So special proteins will be synthesized that will check if DNA is replicated correctly. And it can, so it can slow down this S phase, right? It can slow down to check for DNA damage, or it can completely stop the division of the cells. So those proteins can inhibit cell cycle until DNA can be replicated. So they can kind of put it back to G1 phase, so no time yet, or they can even stop division completely if DNA is not replicated correctly. So during S phase, it's very important to check if cell replicated DNA correctly. Because if we have errors during replication, this is what mutation is. And some mutation can lead to very devastating consequences. Mutated cells with mutated DNA can become cancerous, for example. So that's why we need to make sure that DNA replicated correctly, and either prevent cell from moving forward, return it back to G1 and just wait when it's ready to replicate the DNA. It can slow down the process, or it can stop the process completely. And if it stops the process, that means cells will never really enter this G2 phase. But if cell enter G2 phase, now we have another checkpoint called apoptosis checkpoint. Now, if cell did not replicate DNA correctly and it's entered G2 phase, then we have accumulation. I'm sorry, if DNA replicated correctly, right? If it didn't accumulate correctly, it might never enter this G2 phase. But if it replicated correctly and it's in the G2 phase, we start accumulating special proteins called survivin. What survivin means, cells gonna survive, right? That's a good thing. So if you accumulate lots of this survivin, then cell will proceed to division. So then we will make sure mitosis happens, right? So that's another apoptosis checkpoints. Do we have enough surviving proteins? And if we do, then that means that all these stages passed the checkpoints. Cell was growing correctly, cell replicated DNA correctly, it's entered to G2 phase, so it's continued to duplicate all the organelles and preparing for division, and everything goes right? So everything is good, so we have enough survivin to proceed to mitosis. And then cells start to divide. So it enter prophase, then it enter metaphase, and here we have another checkpoint called spindle assembly checkpoint. Because if we do not assemble spindle correctly, then we might fail to separate chromosomes. And then if we don't separate chromosomes, some daughter cells will get more genetic material, more chromosomes, and some will get less. So for example, some cells might get 47 chromosomes instead of 46, and another daughter cell will get only 45 chromosomes. Or maybe even, worse scenario when we don't split more sister chromatids. So that's why we have this spindle assembly checkpoint. Is a spindle built? Do chromosomes attach to a spindle? Are chromosomes aligned down the equator? And if yes, then cell will proceed to anaphase, telophase, cytokinesis, and divide. So it's very important to have this cell cycle control mechanism that will ensure that cell is healthy enough to divide and it can produce normal functioning healthy daughter cells. And if cell cannot do it, then cell goes through process called apoptosis. So it will die. Now, another important regulatory factor are telomeres. Now, telomeres are located at the end of the chromosome. So here you have a chromosome, right? And at the end of the chromosome, here, on one end and another end, you will have a repeating sequences, T, T, A, G, G, G. So you have T, T, A, G, 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 T, T, A, G, 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 T, T, A, G, 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 right? So many of those repeats. Now, those repeats actually, they don't code for any protein. So why do you need these repeats at the end of chromosomes? Because during every division, the end points of chromosomes, they are damaged. So chromosomes loses 50 to 200 bases after each cell division, right? So so here's your chromosome, right? And everything inside this chromosome, I don't want it to disappear. So here's a chromosome, and inside chromosome you have genes, very important genes that code for your blood cells, for your heart to develop, for your muscles, for all your characteristics. So every single gene is extremely important. But every time when chromosome replicate, part of the chromosome dies. So you want this part to die that doesn't have any genes. So if you don't have this part, that's okay, because you are not damaging genes that is located inside your chromosomes. That's why you kind of lose these telomeres, right? So you lose 50 to 200 base pairs every division. That's a lot. That's why after 50 divisions, telomeres became shorter, and they give a signal cell to stop dividing. So now it's dangerous for your cell to divide, because if you're gonna miss another 200 base pairs, that might be important genes. So you will lose them, you never replicate them, and cells cannot function. Sperm, egg, and bone marrow, however, those cells continue division, and they make telomerase that prevent shortening of telomeres. So if you don't shorten the telomere, then cells can divide longer. And you need your stem cells that make sperm to divide longer. You need to divide to make more eggs. In bone marrow, you make blood cells, red blood cells, white blood cells. So those stem cells need to divide continuously. They cannot stop division after 50 times. That's why they have special enzyme that doesn't allow telomeres to get shorter, right? So that's a good thing. The bad thing is that cancerous cells also can produce telomerase. So that means cancerous cells continue division without any limit. So it's a good and a bad thing about telomerase. So on this electron, or it's an electron microscope, but it's fluorescent dye is used. You can see those bright yellow parts, those are telomeres. So when cells have these telomeres, they're good. Then cells can divide. Well, those not cells, DNA will have the telomeres, right? Just, I'm sorry if I confused you. Cells don't have telomeres. Chromosomes have telomeres. So as long as chromosomes have these telomeres, they can continue replicate the DNA and cell continue to divide. Right, okay. Now, cells receive messages that tell them to divide. Some of these messages include hormones. Now, hormone is made in a gland and transported in the bloodstream to another part of the body, and it exerts a specific effect. So you have hormones. For example, you have thyroid hormone, you have growth hormone, and those hormones travel through your blood and they affect your muscles, your bones, your cells of your organs, and they bring this message for cells to divide and your organs can grow bigger. Right, so that's how hormones regulate division and growth of organisms. Growth factor acts locally. So hormones travel through blood. So it goes from, for example, from your thyroid gland that's located in your neck region, in blood, it can be delivered to bones of your toes. The growth factors, however, act in a local distance. We have epidermal growth factors, EGF, that stimulate cell division in the skin beneath the scalp, for example. Two types of proteins, cyclins and kinases, interact inside cells, activating the genes whose products carry on mitosis. So you have very important proteins called cyclins and kinases, and they promote the division. And so you have them, your cells divide. If you decline the concentration of cyclins and kinases, that might affect mitosis and cells stop dividing. Right, but we said that it's not just important to make new cells, it's important to remove extra cells or prevent cells from uncontrolled over division. So you don't want to make too many cells. That's why we have apoptosis. Apoptosis begins when a death receptor on a plasma membrane receives a signal to die. So your cells have cell membrane. It's like a skin of your cell, right? Of course, it's not a skin in any shape or form, but it's a barrier between external and internal environment of the cell. So on the plasma membrane, you have special proteins called here death receptors. So when they receive signal to die, then some enzymes called caspases are activated. So caspases are inside the cells. When they activated, they destroy the cell. They destroy enzymes that replicate and repair DNA. So cells cannot replicate DNA, cannot repair DNA. They tear apart cytoskeleton. So all these proteins, microtubules, microfilaments, intermediate filaments, everything that make this inner structure of a cell, now that's all gone because of caspases. They abolish cells' ability to adhere to other cells, and they attract phagocytes. Those are big white blood cells that will digest the cell's remnants. Dying cells form bulges called bleps. Membrane surrounds the pieces, preventing inflammation, and your cell dies. So that's, let's look at the picture over here, and then we get back. So this is apoptosis. So here's a cell, right? And this cell, maybe it's not functioning normally anymore. So these cells need to go. So it will start the process of apoptosis. Now, during apoptosis, those caspases inside, they will cause the cell to change its shape, and it will fragment cells into small fragments called blebs. Right, so DNA is gone, cytoskeleton is gone, and cells start falling apart into small pieces called blebs. Plus those caspases inside, they attract these white blood cells that are gonna digest and remove this cellular debris. This process of apoptosis will not cause inflammation. You just remove cells that are not functioning anymore. Example would be a sunburn. So if you have all this skin peeling off, that's an apoptosis of your own cells. Cells were damaged by UV radiation from the sun. They need to go. And that's how they die, through apoptosis. And you will lose this dead cell, and you will regrow a new one. Right? So that's an apoptosis. Another type the cell can die is necrosis. Necrosis is another form of cell death. However, it is associated with inflammation and damage. Apoptosis is a normal physiological process that benefits your body. Necrosis happens when your cells damage and they die in uncontrolled environment, right? And it will accomplished by inflammation, by damage, rather than orderly contained destruction. That's apoptosis. So apoptosis is orderly contained destruction. Necrosis are damage and death with inflammation, with inflammatory processes. So mitosis and apoptosis, right? So again, mitosis when we make cells, and apoptosis when we remove cells that we don't need anymore. So mitosis and apoptosis work together to form functional body. And cancer can result from too much mitosis or too little apoptosis. So if cells divide too much, we can form tumors. Tumors can cause cancer. Or if cells do not die enough, too little apoptosis, then again, more cells are created, and that can result in tumor. Okay, I think that was our last slide. Yes, because this one we already covered, and that's it for this lecture. I want to remind you that we cover cell cycle and mitosis, and we briefly talked about programmed cell death that's called apoptosis. So thank you for watching, and I hope it was helpful.